Welcome to Derms on Drugs, a video podcast brought to you by Scholars in Medicine, the best educational platform in dermatology, and provided at no cost to healthcare providers. Derms on Drugs is where cutting-edge derm meets hit-or-miss comedy. I'm Matt Zires, and each week I'm joined by my residency buddies, Drs. Laura Ferris and Tim Patton, to use our 60 years of combined derm experience to discuss, debate, and dissect the hottest topics in dermatology. It is everything you need to know to be on the cutting edge of derm and to have some fun listening. New episodes drop every Friday on Scholars in Medicine, Apple Podcasts, Spotify, and other major podcast platforms. And don't forget, there is a video component that has the key figures and tables from the articles we talk about. And the episode description has a link that'll get you to the links to the articles and other resources we might discuss on the episode. So let's go ahead and get into it. We've got one of our trademark six-pack episodes, and we are going to go ahead and start with Dr. Ferris. Dr. Ferris, what do you got?
All right. So I have a paper that was recently published in the Lancet. So I thought that this was interesting. It's a clinical trial called Vital. I cannot pronounce like 50% of the words in this. So it is a drug called, a treatment called prademagene zamikeracel. So we're going to call it PZ. And this is for the treatment of recessive dystrophic epidermolysis Bullosa wounds. Okay. So recessive RDEB, recessive dystrophic EB. This is an open label study where the patient was kind of their own control. So this is a phase 3 study. And so first of all, what is RDEB? Little boards review for everybody. It's a rare genetic skin disease. It's caused by a mutation in the collagen 7A1 gene, which is what encodes type 7 collagen. So basically, it's a mutation of type 7 collagen. So that gives you fragile skin. You get these big chronic wounds. These patients are pretty sick. These wounds predispose them to infection, to sepsis, to anemia. And, because of this constant wound healing thing, they get cutaneous squamous cell carcinomas. And these are like really aggressive cutaneous squames that can be lethal. Okay. So what is this drug? So basically, PZ are autologous gene-modified cell sheets. So what they did is they took like 8mm punch biopsies, they made keratinocytes from it, and then they used a retrovirus and transduced it with like wild-type collagen 7A1 gene. So they have your own cells that they've grown up that now have normal collagen 7 in them. And so then what they did was patients had to have at least 2 wounds, and, they picked, here's the treatment wound and here's the control wound. And they only had 11 people in this study who were treated. And so they basically make this sheet, they put it on a piece of Xeroform, and then they go in and sew it in. Patients are hospitalized. The other wound just gets wound care. And then they look to say, what percentage of patients had at 50 and 70% healing from baseline or complete healing. And so they looked at this at week 12 and 24. And so if you look at, complete healing, it was like 16% of the PZ group versus 0 in the control group. But if you look at who had like 50% from baseline, there's about 81%, like 80-ish% at the 2 time points in the PZ group and about 16, 17% in the control group. So that was pretty significant. And then they also looked at pain. So the way that they looked at pain, because it's always hard when you do this with like a control, you're your own control. So they asked about pain during the dressing change, and the pain score went down by like 3 points in the PZ group and by less than 1 in the control group. And that was statistically significant. So, I thought this was kind of interesting. I mean, obviously this is labor intensive and but now we're doing more personalized therapy. And, the other thing is this is a one-time treatment that you sew this on. And then they had patients with, follow-up of up to several years. And so this may be a way to actually sort of fix these wounds in patients. It's going to be a low number of patients. It's a one-time treatment. So I thought that was kind of interesting. We do have 2 other FDA-approved treatments for recessive dystrophic EB. So one of them is called Filsuvez. I don't know why they all have terrible names that are hard to pronounce, but it's a birch bark a derivative, and then the other one is called Vigivec or something, or which is a topical gene therapy, which is basically HSV one transduced with the wild type gene, and so you can like put it on the wounds, and you can basically try to get the wounds to make wild type collagen 7, but you need lots of treatment. So I thought this was kind of cool. It's very, personalized, but disease specific, and, rare but debilitating disease.
This is interesting stuff. I mean, this is what they were telling me in 1996 when I started medical school, that this was right around the corner. And it's only been 30 years, but we're finally getting there.
Yeah.
So,
I mean, now if we can, for how expensive it is to take care of these patients and how awful this disease is, I think it's, interesting to think that we might have more personalized therapy.
yeah, it's very cool. All right,
let's move. Like you said, labor intensive. They actually had to like surgically enlarge some of the wounds because the sheet only comes in one size. It would be neat if they had evidence that you could actually cut the sheet and then you could put it on wounds of different size. They actually went to the OR and debrided them so that whatever ulcer they were treating was big enough. It's almost like they enlarged it, which you would never want to do in an RDEB patient. They made the wounds worse and still had these good results. Really
and
neat.
They didn't debride the control wound. They just, did that with just regular wound care. And they had photos in the paper. If you look at it, the photos are actually kind of impressive. Like you look at it, you're like, wow, that looks significantly better.
Yeah.
Yes. All right. Let's jump on to Dr. Patton's first paper, which has a much less impressive photo to go with it. Dr. Patton, why don't you tell us what you got?
Yes. So I did uncovering the efficacy of a natural homemade sunscreen in protection from ultraviolet radiation by Ince et al., published online June 2025, Journal of Photochemical and Photobiological Sciences. It has what is easily going to win, hands down, the best graphical abstract that there is. I want to say that the authors said, we're not doing this. And the journal was like, you have to. And they're like, fine. And they just made a picture of a guy sitting at a computer using a monitor that has no dimensions of any monitor you've ever seen anywhere, searching on how to make a homemade sunscreen. All right. So anyway, do either of you have concerns about the safety of chemical sunscreens or effects that they may have on the environment? Is that do you?
Maybe a little. I don't know. I don't like the fact that there's detectable levels of them in the bloodstream. I get that, if you look at the oxybenzone or, benzophenone 5 studies, you would need 100,000 times as much to get uterine thickening and all that. I get it all, but I don't know. I feel like I worry about stuff more than I used to. I worry about microplastics. I would have thought that was a joke 5 years ago, but I worry about everything now. So a little bit.
Yeah, and I think that's that's kind of the environment of the world now. Vaccines are bad and pasteurized milk, and so chemical sunscreens are kind of falling into that same.
Okay, I don't believe in either of those things.
I know, but when you get on the wellness bloggers, that it's unfair to say they're all like that, but a fair number of them are like, this is why I do homemade sunscreens. This is why I'm not getting vaccinated. This is why I don't know, who knows what else. I have zero concerns. I don't look at my sunscreen ingredients. I don't care. I put whatever I put on. It's not so. I'm not saying if a patient comes in, I don't say, oh, you're being silly. I just tell them personally, knowing what I know about all of the data that's out there, none of this stuff concerns me. But if you do have these concerns, and then I kind of point them to resources where if they have those, but some of those resources are homemade sunscreens, right? So there was an article from 2020, 2020, in the Journal of Cosmetic Dermatology, looked at 15 different recipes for homemade sunscreens, and the SPF and the PF UVA were pretty bad. They weren't in vivo studies. It was testing done on these polymethyl methacrylate plates. And most sunscreens that were tested from this homemade recipes have SPFs in the 1 to 2 range. The best one had an SPF of 5. But, who knows how those numbers would relate to actual damage in the skin. But the current study compared the effectiveness of one particular homemade sunscreen homemade sunscreen to a commercial product. In this case, Banana Boat Dry Balance SPF, which has all chemical sunscreens: homosalate, octocrylene, octisalate, and avobenzone. And what they did was they compared the homemade one to the commercial product on skin samples. So they were taking skin from surgeries that were done. They had leftover skin. So they took these little skin samples, they put on the sunscreen, subjected them to UV radiation, and then they did histologic exams of UV-induced damage. So figure 2B shows how each sunscreen reduced the number of sunburn cells. So they all reduced them compared to, no sunscreen at all. And commercial sunscreen was better than the homemade sunscreens. The difference in reduction didn't reach statistical significance between the 1 day old homemade sunscreen versus the commercial sunscreen. If you look at the graph, it's a huge difference, but I guess it just didn't reach statistical significance.
Fair.
All the other studies, like the homemade sunscreen actually was comparable, which, that earlier study, if you have SPFs of 1 to 2, I mean, they cooked these skin samples. It was 20 joules per centimeter squared. I think if you're outdoors at midday for 40 minutes, you're at like a little bit above 10 joules. So they absolutely cooked these skin samples under this light. And when you look at the other things like the cyclobutene pyrimidine dimers, that's sort of the footprint of UVB damage, there wasn't a huge difference between any of the sunscreen studied. For the homemade sunscreen, they did 1 day old and 3 week old. They also looked at this 8-oxo-7-A dihydro-2-deoxyguanosine. They shortened it to 8-oxo. That's your sort of UVA footprint damage. All the sunscreens were comparable. I would have expected that to be a lot worse. And I question, maybe doing it in that lab setting with a little teeny skin sample that you're able to put a decent amount of the sunscreen on. That's not going to correlate to how you're using this in the real world, where you're slathering on a much larger area. So in real world use, would this data be a lot worse? One would think, but I was kind of surprised that it wasn't as bad as I expected it.
So you're going to start telling patients to make homemade sunscreen?
No, because, and this is a lot from if you talk to the chemists, or I watched a few videos on chemists. I went back to the Lab Muffin Sciences with Dr. Michelle Wong. It is really, really hard to make topical medications and to make them effective where you don't have to worry about them getting contaminated, where you have controlled and have dispersed the active ingredient across the thing that you're putting on with these high shear mixers. There was a blogger, a different chemist blogger, and for 1 year, she tried to make her own sunscreen and then do the legitimate in vivo SPF, minimal erythema dose testing. And she said it was so hard, and sometimes things that didn't make sense. One time she changed the fragrance and everything else was the same, and the SPF went down. And she's like, that doesn't make any sense to me. I think that these companies are very highly regulated. They go through rigorous testing to be able to come out to the market and say what they say. And sitting at home on a stove using non-nano sized UV or zinc oxide powder, there's just no way that these could be anywhere near as good. If patients have the concern, you just point them towards the mineral or the inorganic sunscreens that are out there, and they have lots and lots of choices. So
yeah, for sure. That makes sense. I mean, so are people buying powdered zinc oxide on Amazon and throwing it into some CeraVe and calling it sunscreen?
That's one thing that they do, yeah. But a lot of these recipes are these natural seed oils and other oils and beeswax pellets and all these other things that they say also have antioxidant properties, and that's going to protect you from. That would be mostly UVA damage. UVA is the one where it's sort of indirect damage through these reactive oxygen species. So, no, I wouldn't do it. I mean, it's I don't know. I'm just lazy. It's like everything else. You can make your own beer, and I've done that, but Man, just buy it.
Well, with the amount of beer you drink, that would be a lot. You have to quit your full-time job.
That's
So I
okay.
get it.
I didn't think we'd make this personal, but okay, fine.
All right, let's.
So wait, can I ask you one more question on the
Yeah.
sunscreen? What do we think? Polypodium leukotomas, heliocare. Anybody believe in that?
Oh, definitely works.
I think it protects you from UV induced damage.
Yeah, like they have decent data, right?
Yeah,
like MED testing data. Okay.
There's other like if you go to like integrative dermatology sort of talks about natural things to do to protect yourself from potential sun damage. There's a fair number of things that are very little harm. Is it going to replace sunscreen? I wouldn't take a polypodium leukotomas and not put any sunscreen on, but I think in conjunction with everything else that we do to protect ourselves from the sun, it can help.
The polypodium leukotomas is also interesting. I mean, there is some data that if you increase the dose, you increase the protection from sunburn. But I there's not any evidence I've seen that it has doing that makes it more protective against, say, skin cancer, which is the real concern.
Or photoaging, also a big concern.
Yes.
Yeah, there's actually one study showing that if you added it to like narrowband and maybe a topical JAK inhibitor, it improves the treatment of vitiligo. So I thought that was kind of interesting. So sort of decrease of reactive oxygen species. So, I'm hoping someday we're gonna have something that's like an oral medication that can at least provide some sun protection or, some of the benefits of sunscreen. So interesting. Cool.
All right, so let's jump over to my first six-pack article, which was updated expert opinion guidelines regarding the effects of immunosuppressant agents on patch testing. So first, let me just summarize kind of what the recommendations are here. For the topical medications, for the area you are going to put the patches on, minimum of 7 days for anything. That you want none of it applied. And if we're talking high potency topical steroids, so clobetasol, you want at least 14 days between applying the steroid and using the patches. Now, this was an expert opinion, so there was a big range. I mean, there were some members of the North American Contact Dermatitis Group said, a day is fine. Other people said 14 days, but they sort of came to let's vote and see what we come up with. And in the absence of the kind of studies it would take to figure this out, this is the best recommendations we're ever going to get. And then for systemic meds, for systemic steroids, the recommendation was that people are off for 7 days. The recommendation for intramuscular triamcinolones, at least 30 days between a shot of that and getting patches put on. Then most of the other drugs that we don't use much of anymore, methotrexate, Imuran, cyclosporin, Cellcept, all of those got 7 days. Oral JAKs got 7 days. UVB phototherapy, 7 days. Biologics for atopic derm, it said 14 days. But there were two asterisks there that said you probably don't need to hold these. So there's conflicting evidence that they may affect the results of patching to some antigens, but not all. But really, the data that's out there suggests they probably don't really affect patch test reactions much at all. And so the article does have a footnote to that effect. Psoriasis biologics, you don't need to hold. Antihistamines, you don't need to hold. And then the other thing is, well, if you can't stop drugs, because if they're just flare so badly that you can't patch test them, generally you can get a pretty accurate patch test on less than 10 milligrams a day of prednisone, less than 15 milligrams a week of methotrexate, less than 3 milligrams a day of cyclosporin, Cellcept, less than 1,500 a day. So biologics for atopic derm says if the biologic cannot be held, patch tests can still be performed. Biologics may selectively impact patch test results of some allergens. And then for an oral JAK inhibitor, the patch test can be performed. And the way that I really think about it with JAK inhibitors is if they're on a low enough dose that they're getting some rash, then it means that the dose they're on is not enough to suppress. If the rash is being caused by contact derm, the dose they're on is not enough to suppress it. And so you should be able to get a positive patch test to it. So
It should not be the case for every single drug. Why just JAK inhibitors?
To a very significant degree, that is how I think about it. That if somebody's only partially controlled by a drug, it is very likely that you get a very accurate patch test with it. We just don't have a lot of data to really back that up. But from a perspective of just what's common sense, that makes a lot of sense. So not a ton to really talk about there, other than, it's really useful information. I want to get it out there to everybody because that's a question that comes up frequently. Any other comments from the two of you?
No, I like the minimum numbers that they give there, right? I mean, if the patient's miserable, you could say, all right, well, let's take it down this low, and you can still get patch tested on that, and it's going to be relevant. It's useful.
Yeah. Okay. All right, let's move on to our next one. Dr. Ferris, what do you got?
Okay, so this is from the most recently published in JAMA Dermatology from Emily Wenande et al. Human HPV and actinic keratosis burden, the VAX-AC randomized clinical trial. So this was done in Denmark, and it was a randomized trial, 1-to-1 of like 70 patients who were immunocompetent adults who had 15 or more AKs. They were randomized to either get Gardasil 9, which is a 9-valent alpha papillomavirus vaccine, or sham vaccine for 3 doses. And so what they looked at was the percent reduction in baseline AKs at 2, 6, 9, and 12 months from the first vaccine on. And then from there, they looked at like total AK numbers, thick lesions, and new AKs over a year. So, at baseline, looking at these patients, as you can imagine, most of them, because they have AKs, are patients who are white. However, there are actually a lot more type 1 skin patients in the vaccine group and more type 3 skin patients in the control group, which you could imagine could have an impact on AK number over a year. Also, the patients in the control group had more skin cancers in the previous 3 years, but they both had a similar number of AKs at baseline. And so the other thing to know is that they said, due to ethical concerns, we could not leave AKs untreated for a year. So they both had cryotherapy basically to any Olsen grade 2 or 3 AKs in the test area at months 6 and 9.
Okay.
So there was some cryo, so not like the cleanest thing in the world. But basically, reduction in baseline AKs, basically the HPV group had a higher reduction. So at month 2, 35% reduction versus 25% in the sham group. Month 6, 47% versus 29% in the sham group. At a year, 58% versus 47%, although the number, I was like, the number's wrong in the table. We'll have to send a letter to JAMA Derm.
Yes, I saw that. It said 4% and it meant 40%.
4.007%. It's 47%. And then the median number, like total number at the end of the observation, was lower in the HPV vaccinated group. So it looked like they got fewer new ones. They also had like fewer thick AKs. Now, did they have fewer skin cancers? No, they did not. It did not, that over a 12-month period. That's a short period of time, but they did not. So, what do we think? So based on the sort of percent reduction in baseline AKs, there's maybe a 10 to 18% difference in the vaccinated versus sham group. So, maybe this is helpful. Maybe it's not. They said maybe this could be adjunct therapy to AKs. I know. Other things. Patients were told to use sunscreen, right? That alone can treat some AKs. It's hard to count AKs. I've done like 1 or 2 AK studies, and it's like hard to count discrete AK numbers. They didn't test for HPV in their AKs, so we don't know. And the data on HPV within AKs is more like I think that's more of an issue in immunocompromised patients, but these were all immunocompetent. And then the other thing was that 76% of patients actually got field treatment outside of the treatment, outside of like the target area of the face or whatever. So, could getting PDT or 5FU or a imiquimod on your arms impact your, could you have an abscopal effect on your face? We don't know. So
I thought that was so bizarre. They said because of ethical concerns about leaving photo damaged people untreated. Like, that's not an ethical concern.
No, I thought that was like a little over the top. Like
for
that
a period of 6 months, they're going to be okay.
We call that like the whole state of North Carolina is photo damaged people untreated for 6 to 12 months. So yeah, I thought that was, I get like we will biopsy things that look like that are suspicious for skin cancer, but I think that they sort of made it hard to find a meaningful endpoint. So I don't know that we're going to start doing HPV vaccines. It doesn't really make sense that HPV vaccine much later in life to something that makes antibodies to capsid protein should make a difference in established HPV disease. But
you'd have to have decreased number of cancers as your endpoint, right? Decrease in the number of AKs. Fine,
but who cares?
Yeah, if in the end you're not decreasing the number of skin cancers that these patients get, that ought to be like the only thing that concerns us.
I agree. Yeah.
Agreed.
So I thought it was worth the discussion because I think people would be like, oh, maybe we should be doing HPV vaccines. I didn't think it was enough for me to say I'm going to even think about this with AK patients.
I'm like so happy that the paper exists because, right? You're kind of thinking, what can we do for those horrible patients? But a lot of those patients are also immunosuppressed, and that wasn't the population studied either. So,
right.
Yeah. All right. Next paper, Dr. Patton.
Treatment algorithm suggestion for Hailey-Hailey disease by Giannopoulos et al. In the June 2025 Journal of Cutaneous Medicine and Surgery. Just a nice quick review, Hailey-Hailey treatments, including conservative measures, topical agents, systemic agents. And they basically came up with a recommended algorithm that I thought was pretty straightforward. So the algorithm's there, and I think that makes sense. Before I talk about the paper more quickly, what's like my take on Hailey-Hailey is patients usually come in when it's pretty flared. It's pretty, it looks pretty impetiginized and infected. I typically swab the areas, and whatever grows out, those are my systemic antibiotics. And when Hailey-Hailey patients flare, that's what I use. And I'll include a viral culture in there as well. That's how I think of here's how I'm going to keep a fair number of my Hailey-Hailey patients under long-term, I don't want to say control because they still flare, but that's kind of it. I don't know that I have like a maintenance for these patients. They call when they flare and I give them whatever the antibody cocktail was that I was able to culture.
Whatever the antibiotic, not antibody.
Sorry. Back on my immunobulla stuff. Do you guys manage Hailey-Hailey any differently?
I do not culture them. I can't remember the last time I cultured somebody. I think if they look impetiginized, I kind of just default to doxycycline. Maybe I should be culturing them.
Like sometimes you get a gram negative, and then you like to see what the sensitivities are. I have found it to be helpful, but yeah, you're right. Empiric therapy may be enough.
And then do you repeat culture them every time they flare, or do you kind of treat them according to that original culture?
I treat them according to the original culture, and if they didn't respond, I would have them come in. Because a lot of times they send a message. They're like, pretty sure my Hailey-Hailey's flaring. I'm like, okay, we'll start these, and then let's do a follow-up in a couple of weeks.
Okay. So recommendations beyond antibiotics, anything maintenance? Like do you do acitretin? Do you do...
Not routinely, because I have felt that between flares, they're fine, and they're not going to take it. I can't, I don't have hundreds of Hailey-Hailey patients, but it's not like I've never not seen, I've seen it. And that's like that sort of management plan, that seems to work in my experience. I may be completely not recognizing everybody that I'm treating poorly. That seems to do fine.
So basically, systemic antibiotics whenever they need them. Do you even do TCIs, like maintenance therapy? Kind of nothing as long as they're.
If I do maintenance, I would probably give them a steroid and say use this a couple times a week when you're not active. But again, like I don't know if they're going to do maintenance if they have tons of flares. But I've just been lucky with most of my patients. Some other so the algorithm, like I said, is pretty straightforward. Some of the treatments not shown in the algorithm but discussed in the article I thought were worth talking about really quickly. Oral anticholinergics, because they do have Botox in the algorithm. So for frequent flares, control sweating, maybe sweating acts as a trigger for the onset of disease. Sure. Oral apremilast was mentioned. I think they pulled out like 16 patients reported in the literature, 10 with improvement. Dapsone, but it was only a couple of cases. Immunosuppressants like cyclosporin, methotrexate, thalidomide. I have not had to do that with my Hailey-Hailey people. Apparently, Dupy had some good results. Article of 11 patients treated with Dupy, an improvement was seen in 10. So maybe it's a TH2, like a lot of things are turning out to be. They touch on ablative techniques, dermabrasion, CO2 laser, photodynamic therapy. I would throw in superficial radiotherapy only because I have an article that I published on treating a Hailey-Hailey patient, and she was recalcitrant. We had
Wait, I
kind of
thought you
done
had
all those things.
Derriers.
It was both. I've done both.
Oh, okay.
Yeah, Derriers, we had the patient with the full body. This was a patient with Hailey-Hailey only in a few areas, and she got electron beam therapy. Wasn't mentioned in the article. It worked really well for her. There were also a couple other case reports demonstrating the effectiveness of superficial radiotherapy. So if you have a radiologist that's willing to kind of treat those patients, I
A
think that would
radiation
be
oncologist, right? It's a
yeah.
radiation.
What did I say?
Yeah, radiologist. They can CT scan
Yeah.
them, but
Yeah.
they won't.
Radiation oncologist, they could do the superficial radiotherapy. Worked really, really well for that patient that we published on.
Okay.
So how about do you think about this like Derriers? How do you think about it differently than Hailey-Hailey?
It's it's kind of a similar type story. Although I don't culture my Derriers. Derriers, I do think like maintenance acitretin. I have my Derriers on that stuff more frequently. Hailey-Hailey does seem to be, again, for me, bacterial, flaring it and treat the whatever's flaring it. I don't think of Derriers as the same way of like flares are caused by this. Derriers, those patients have disease like in between flares where Hailey-Hailey, in my experience, doesn't.
All right. Let's move on to our last article here. And this was one that I chose just because it was really interesting and something that was just fascinating to me. So the title of the article was JAK inhibitor withdrawal causes transient pro-inflammatory cascade, a potential mechanism. And they think this might even be related to MACE events. So essentially, here's the idea that whenever and we're all kind of familiar with the idea that the way that JAK inhibitors work and the JAK pathway works, the two sides of the cytokine receptor come next to each other. That brings two JAKs next to each other and they phosphorylate each other, and that puts them in an active state. And in that active state, they then bind ATP and transfer it to STAT, activating STAT. Essentially, all the JAK inhibitors that we have, with the exceptions of ritlecitinib and deucravacitinib, which is not really a JAK inhibitor, they bind to the site that binds the ATP that's going to transfer it to STAT, but they only bind that site after the JAK is activated. So the JAK is it's activated itself. It's been phosphorylated. So that opens up its you can think of it, that opens up its binding site for the other phosphorylation. Well, when that opens up, that's when the JAK inhibitor binds to it. And so it sort of locks the JAK into an active state. And so as you are on the JAK inhibitor, you're accumulating this JAK enzymes that are sitting there locked in the active state, but unable to do anything because the inhibitor is blocking the site that needs to bind the phosphorylation. Well, if you then miss a dose or stop the drug, as it starts to get cleared intracellularly, as it comes off, you now have all of this JAK that is built up in the active state. And so you get this almost like little spike of JAK stat activation. And that may be why we see people who will tell me like, oh, if I miss a dose in the morning, that night I can already tell I'm starting to flare and get itchy, to which I was always like, man, that is like crazy. Like you usually don't even see that with like prednisone or something where somebody flares that quickly. And so this was just fascinating to me mechanistically, the idea of how this might work. And so that, does it mean anything terribly clinically relevant? Not sure. Maybe, we should be really telling our patients, hey, try not to miss any doses. Because it could cause you to have a quick inflammatory reaction. But I just thought it was really cool mechanistically, the idea that we're sort of accumulating the JAK in the active state intracellularly. Just thought it was cool. That's it.
Think there's any danger in that? Like, could this make people more likely to have like thrombotic events or anything?
They suggested that was possible, that there's an increase in urokinase and an increase in interferon that are part of this, and that might be related to the thrombotic effect. But, as you guys have probably heard me talk about, whenever I really look at all the data, the JAKs that we have in dermatology, there's no data that suggests that there's actually an increased risk of any of those things. Now, is there an increased risk of those things? I suppose it's imaginable, but we've got a lot of data now around the JAK inhibitors in derm, and there really hasn't been any indication of a MACE or VTE signal. Remains the case that they're immunosuppressive drugs, so that is absolutely the truth. But otherwise, nothing that really is highly suggestive to me.
So should we be tapering JAKs instead of stopping them?
It's a really interesting question, like a really interesting question, right? And it, how are you going to taper? Like go down to 15, and then like you can't do a half a pill. Doing every other day wouldn't make any sense. So it's
so quick. You're saying it's like
yes,
that day. Okay.
yes. So doing it every other day, you might like just be causing this reaction to happen every other day. It's really interesting. We don't know what to make of it. We don't know if it means anything in that is clinically relevant, other than this may explain why people seem to relapse very quickly. At least some patients seem to relapse very quickly if they even miss a single dose. So that's it. Just thought it was cool and interesting. And with that, we're going to close today's episode, one of our shortest ever, which is very impressive for us. And so I want to thank everybody for joining us this week. If you got questions, comments, or ideas for topics to cover on the show, shoot us an email at questions@dermsondrugs.com. Hope you learned a few things. Hope you laughed once or twice. And mostly, we're hoping you're planning to join us next week. Until then, I'm Matt Cyrus.
Tim Patton.
And I'm Laura Ferris, and we are Derms on Drugs.